All right, so thanks for the introduction, Teresa. I'm Holly Peay from RTI. Cynthia and I are going to tag team this, so I'm going to get us started. Cindy's going to jump in a few slides in, and then I'm going to pick it up for the end. It's a little more difficult to do this virtually, so we will probably need to ask for questions at the end, but if there is any way you can make yourself known to us, if you have any burning clarification questions, we can try to answer those as we go. Otherwise, we'll just hold them till the end and see if we can address them all then. So I'm going to go ahead and advance my slides. We're going to talk to you about an expanded newborn screening study that we've been, that went too far, conducting in North Carolina. Many of you have probably heard about the Early Check project already. I hope that's true. If not, we're going to give some overviews, but we're going to really focus today quite a bit on the screening panel and the procedures we use for screening and follow-up, as well as talk to you about some of our screening and follow-up outcomes. We're going to talk about some of the challenges and opportunities that are inherent in conducting large-scale research, which has been fun and challenging. And then we wanted to tell you about some of our next steps that we're planning for the future of Early Check. That's the agenda for today. I'll do a bit of a partners and funder disclaimer in the same slide. Those of you who know RTI know that we're an independent research organization. Those of you who don't know us may not know that we were actually developed to collaborate with the academic and research institutions surrounding, especially our immediate geographic area in the Research Triangle. We conduct clinically oriented research at RTI, but we don't do the clinical engagement. And Early Check is actually about the most clinical that we get. So I'll talk to you about that, but we really rely on our partners, and Cindy is here from UNC Chapel Hill. We also have collaborators from Duke, from Wake Forest, and really importantly for the success of this project, from the North Carolina State Laboratory of Public Health, which is a really vital collaboration that I'll describe a bit more as we go. We have a broad range of funders. This really is a public-private partnership sort of model. The underlying funding for Early Check to actually get Early Check started, and we continue to have funding to implement Early Check through NCATS, through the NIH. We also have NIH funding from the Child Health and Human Development, as well as funding from advocacy groups and industry. So we have a broad set of disclaimers on the screen. So a little bit of background about Early Check. It is a research study. It is conducted across the state of North Carolina. And the study was designed to develop and evaluate methods to offer voluntary screening for conditions that are not currently part of newborn screening conducted in the state of North Carolina. In North Carolina, we have about 120,000 babies born a year. So we have a very large participant pool who are eligible for this study. The primary objective of Early Check is to acquire data to inform policy for newborn screening and to inform nominations or lack of appropriateness for nominations to the recommended uniform screening panel. We also do quite a bit of exploratory and assessment work to understand parents' interest in screening for new conditions, to figure out how feasible and acceptable the systems we developed are to implement screening, and to provide services, of course, for screen-positive infants. The project was also envisioned as a foundation, and we'll talk a bit more about this when we talk about our future work, but really we intend this to be a foundation for long-term research and really helping to understand the framework by which new conditions could be added or not to the RUS, but also to envision a future where states could offer a voluntary panel of non-RUS conditions that would be used for offering screening in a voluntary manner across the state. So I'm going to do a overview of our flow, of the early check flow, but we're going to get into more detail later. I think you'll see this and be like, oh, it's not very specific, and I want more information about each of these boxes, and we'll get there to most of it today. So we conduct recruitment in as part of our really important collaboration, as I mentioned before, with the state. We actually have access to contact information for the parents whose babies have newborn screening in the state, and that is what makes our recruitment possible. So we actually reach out to all parents whose babies have newborn screening in the state and invite them to early check. We have other recruitment approaches that if we have time and people are interested in, we can talk a bit more about. But importantly, everyone receives a letter from Early Check, and also it's on the state letterhead. So that letter goes to all parents who give birth in the state. There are different models for parental permission. Most of our parents, though, give permission online. So there is an online portal, and we'll go into that in a bit more detail as we go through this presentation. The screening test is then conducted. Of course, this is newborn screening for rare conditions, so the large majority of our parents get a normal result, and those normal results are reported to them through the research portal. So unless parents contact us, all of this is done electronically, and that has been an interesting learning process that we can talk more about if folks are interested. But for the parents for whom their infants have a positive result, they receive a contact from the study genetic counselor, and there's a series of steps that we go through for confirmatory testing that we'll talk about in more detail for each condition and for diagnosis. And then we do care coordination support, and then there are follow-up steps as well that include providing information and support to families longer term, doing developmental surveillance, and in some cases doing some interventions actually for child development. That's our overall process. So I'm now going to hand it over to Cindy to talk about our first, one of the three conditions we have on our panel, we've had on our panel historically. And Cindy, just let me know when you want me to advance.
Okay, thanks, Holly. So we began SMA screening as part of the Early Check program in October of 2018. Subsequently, North Carolina is screening for SMA as part of their standard state newborn screening. Next slide. This is the algorithm that we used for the Early Check SMA newborn screening. We utilized a real-time PCR assay to quantitate the SMN1 exon 7 copy number, and abnormals are those with no SMN1 exon 7 present. If one or more exon 7s were present, that was a negative screen. For those that had 0 copies of SMN1, based on the screening, those were reported out as positive screens. The RTI team would notify the UNC genetic counselor, where we did the follow-up and confirmatory evaluations for those patients detected. And then there would be, the infant would be seen on the same day by a geneticist, a neurologist, and the genetic counselor, and blood would be obtained for confirmatory testing. Next slide. And while it was, critically important to be able to return results quickly, we also asked for additional follow-up of those families through developmental and family assessments. And some of the follow-up, such as the confirmatory testing, was done as part of the clinical evaluation and not part of Early Check. Next slide. I just wanted to show that I'll talk about each of the 4 cases that were picked up through Early Check, but the cutoff was formed through use of some standards that were obtained through other states and the CDC when we were setting up the validation for the newborn screen. And you'll notice that just above the cutoff there, where it says false positive screen, that will be denoted as our case number 2. and it was just above the cutoff. Whereas you see more on the upper left side, the true positives were well above that, that cutoff. And I'll talk a little bit more about that. Next slide. So to go through the cases that were identified through screening, there were 4 cases, 3 were confirmed, and 1 was found to be a false positive. Case 1 on this list was an infant whose mother and father were known to be carriers for spinal muscular atrophy. The mother's brother was affected with spinal muscular atrophy, and the parents had previously undergone carrier testing. They with their first pregnancy, they had amniocentesis that showed that the fetus was a carrier for SMA unaffected. That child subsequently was born and did well. For the second pregnancy, the mother was given the option of having non-invasive prenatal screening done. And not realizing some of the limitations of that screening test, but knowing that it was less invasive than amniocentesis, they decided to do the NIPS screening. And that company, which will go unnamed, they reported out that the fetus appeared to be unaffected. They decided to not have any additional confirmatory testing done prenatally, but fortunately signed up for early check by the time the baby was born. So this case was definitely, above the cutoff. We saw the infant for confirmatory testing and found that indeed he had 0 copies of SMN1 and had 2 copies of SMN2, which we don't have enough time to go through all the genetics of SMA. I'm sure many of you are familiar with it, but the number of copies of SMN2 can tell you in some respects what the clinical course will be. So typically having 2 copies of SMN2 would be a type 1 spinal muscular atrophy. And this infant did receive their first loading dose of Spinraza at 30 days of age. The AAV9 titer, which is measured to see if the infant would qualify for gene therapy, that was positive, but eventually went down, and the infant did receive Zolgensma gene therapy at 8 months of age and has done well. The second case was what we say is a false positive screen. Interestingly, this infant had also had a borderline severe combined immunodeficiency newborn screening test through the state, but the second sample was negative for SCID. The baby was hospitalized for persistent neutropenia soon after birth, and that's likely what caused this borderline elevation, so that it was picked up through the screening. The infant was subsequently diagnosed with Shwachman-Diamond syndrome, and the mother is a SMA carrier. The father is not. The baby was found to be an SMA carrier. The third case, this was an infant born prematurely who did have confirmatory testing that showed 0 copies of SMN1 and 3 copies of SMN2. And this child received their first dose of Spinraza once they got to their due date, since they were premature. And the AAV9 titer has been positive. And I think eventually went down, and that baby did get Zolgensma. And then case 4 also confirmed to have 0 copies of SMN1 and 3 copies of SMN2, and was able to get Zolgensma. The initial AAV9 titer was negative, and Zolgensma was administered at 7 weeks of age. Next slide, please. And that last child was the first infant to receive Zolgensma at UNC, and which prompted this report in our local newsletter. Next slide, please. So as Holly mentioned, we've also been doing screening for the FMR1 gene, both full mutation and then pre-mutation. The pre-mutation for Fragile X is offered as a second consent. So we realized that there were certainly more ethical issues in doing screening for the pre-mutation for Fragile X, but there's a longstanding interest for Fragile X research that Don Bailey, the overall PI for early check, has had. And so it was really Don who got interested in newborn screening because of his interest in Fragile X syndrome. So we began a Fragile X screening in October of 2018. Next slide. For the full mutation, the genetic counselor would contact the family and the pediatrician, and there were options of scheduling a visit in the in-person early check clinic versus having a telegenetic counseling session. Most of the families opted in for the telegenetic session with the genetic counselor. A saliva kit would be sent to the home to provide DNA for confirmatory testing. Also, there would be an initial developmental assessment, either directly or by parent report. And then the families would be asked to consent for follow-up of that infant. The clinical long-term follow-up was done with care coordination and some special types of intervention. Also, testing other family members would be offered depending on the sex of the proband. Either one or both parents would be offered. I'm sorry, for the full mutation, the mother was offered testing. Next slide, please. And then for the pre-mutation, it followed a similar stepwise process. Confirmatory testing would be done through saliva kits, and depending on the sex of the baby, one or both parents would be sent saliva kits. And then those families were also asked to participate in a long-term follow-up with the early check program. Next slide, please. So of the 16,881 babies who were screened, some of the specimens were unsatisfactory, mainly due to insufficient quantity left on the dried blood spots, because early check is done after the state completes their standard newborn screening. And so, sometimes there just wasn't enough blood spot left. In 7 cases, they were unable to amplify the DNA. For full mutation Fragile X, there were 4 positive screens and all were subsequently confirmed. 2 were males, 2 females. One case was the first known case of Fragile X in the family, and 3 of the families did have a known history of Fragile X syndrome. For those cases that the parents consented to receive results of pre-mutation testing, there were 46 positive cases, 21 males, 25 females, 30 were confirmed, 10 declined or didn't send a sample for confirmatory testing, and some are still outstanding. And in 25 of those cases, there was no known family history of Fragile X. And then we did identify cases with the pre-mutation for those that had not requested it, and so we did not report those out. There were 38 of those. For the parental testing, the mothers followed up more often than the fathers did. but 19 mothers were tested. 12 were identified as pre-mutation carriers. 3 had a gray zone allele size. 2 fathers were tested, and 2 fathers were confirmed as pre-mutation carriers. And in 2 cases, they declined testing, but because of the sex of the baby identified, they were presumed to be carriers. Next slide. So all of the information was conveyed to the families, and all with the pre-mutation have participated in the follow-up developmental evaluations and family assessments. Next slide. For the pre-mutation, it's varied a bit in terms of those who are participating in the follow-up. There is a great interest, and some studies have shown that there can be subtle effects from being a pre-mutation carrier for Fragile X. And some of those families are, have been willing to participate in that follow-up developmental and family assessments. Next slide. I'll turn it back over to Holly.
Okay, so I'm going to give a quick overview of Duchenne muscular dystrophy, which we added in November of 2020. This is our newest condition. We actually are using CKMM screening, which I'm going to show you a bit about. Many of you may be familiar with this. There's actually an approved assay from PerkinElmer that detects CKMM, which is a specific muscle component of CK in dried blood spots. And it's a biomarker of muscle damage. So it is not specific to Duchenne. It may be increased in other neuromuscular conditions, but also due to environmental factors. So you will not be surprised as we go through to see that we have a much higher false positive rate than we do for the other 2 conditions based on the type of screening that we are conducting. So this is a tiny slide and you may not be able to see it very well. I'm sorry about that, but it shows our cutoffs. We actually intentionally have our cutoffs for CK to be quite low. And that is because part of the objective of this study was to understand what else we would find if we use CKMM screening. We intentionally set our cut to start with below what we anticipate any newborn screening study would, or newborn screening public health practice would implement. Again, so we can understand what other kinds of conditions that we might find, but also to really try to make sure that we're keeping our, that we're not missing cases. So we anticipate we're actually going to raise our screening threshold at the end of this calendar year. Right now, if people have CKMM above 1626, that's a positive screen. There is a similar kind of flow that we've heard before, except that after the telegenetic counseling, we actually ask parents to consent to 2 things. One of them is a latent CK, or a second sample that's collected after the baby's birth to look at a second CK level. And the other one that we do at the same time is a next-gen panel of 86 conditions that can also be done on the residual dried blood spot from the state lab if it's sufficient, and if not, we get a second sample. And so we actually get those, we get the latent CK much faster because it's a faster test, but we use both of those sets of data together to determine what sort of follow-up is needed. The clinical follow-up for these children is done at Duke with Dr. Eddie Smith. And we have a bit more flexibility in this flow in terms of how parents engage with us and with Dr. Smith. Most parents to date have had a telegenetic counseling visit with our telegenetic counselors, but also with Dr. Smith to do some kind of anticipation and preliminary guidance, and then have a 6-month actually in-person clinical visit with Dr. Smith. So we have screened at this point 6,000 plus babies. We've had some unsatisfactory, as Cindy mentioned before. We are using the residual dried blood spots, and so sometimes we simply do not have enough, or the quality isn't good enough. We have had 42 positive screens. This number keeps growing, so it's actually higher now, but as of the end of last month, you see that we have most of those are females, are males. 34 of those parents have consented for the next-gen sequencing panel. We had 7 who did not consent and 1 who is outstanding. And on 29, the panel is complete. The panel is pending. The genomic results are normal in 24, but we have found 1 individual with Duchenne, but you see we've also found some interesting other findings. So we have a variant that so I'm looking at the second case here in CLCN1. That individual actually, we think the father is affected as well with very mild symptoms. It's just a very mild muscle mass increase and sometimes having some problems with stiffness in the father, and that is anticipated to be a similar phenotype in this male infant. We've identified then as well 2 carriers and an individual who is heterozygous who might have a pathogenic variant in COL6A1. These individuals are all very young, and so we are still waiting to do the clinical follow-up on all but the first individual we identified with Duchenne. We've also done latency K in 30 of these infants, and in a couple of them, we've had insufficient specimens and the parents did not want to repeat those. We had 25 in the normal range. We had 1 that was slightly elevated. And in that case, we report variants of uncertain significance to our team and to the clinical team at Duke. We have 2 that are pending. You will probably note that the individual with Duchenne did not have a latency K done. The family declined that, and so therefore we do not have a second specimen in that individual. So we have a sort of a different situation here because we have, as we indicated before, a much higher false positive rate. And so the way we communicate with these families is quite different. And we are, as you would imagine, seeing that the individual with Duchenne, for example, had a CK level that was above the threshold for detection. So we're able to give some more anticipatory guidance to families at the initial screen positive call about the findings that we've had so far and where we think their child might lie in those findings in terms of the potential for a true positive to be indicated in that baby. We therefore, are more reassuring than we would be for the Fragile X and the SMA screen positives about next steps for those parents. We have lower rates of individuals who have participated in the follow-up genetic counseling and the developmental family assessment. But what we're seeing with these families is many of them are taking a little break and then they come back to us. So also our timelines have been spread out more than we've seen for SMA and for full mutation Fragile X. So I'm going to just spend now 2 or 3 minutes talking about some of the challenges and opportunities in large scale research, and then we'll close and ask for some questions. So you've seen that we have almost 17,000 people enrolled. You see a little map of the state here. You'll probably have good enough geography to see that we move into South Carolina. Parents whose babies are born in North Carolina, even if they live in South Carolina, they have newborn screening in North Carolina, and they are eligible for early check. And so we do have some families down here in South Carolina, but we have parents who enroll their children in 99% of our counties and in all of the birthing hospitals in North Carolina. So we have a very large and actually quite diverse group of participants, which is great, but comes with some challenges. One of the challenges just practically for our team is to maintain the really kind of intensive effort it takes to continue to recruit people at the same level over a very long period of time. So you see here our recruitment is both pre and postnatal, starting when we initiated screening in October of 2018 all the way to August of 2021. We're trending up. We've actually done pretty well during the time of COVID because of the way we've run our study. But it takes an enormous amount of effort to get messaging consistently across the state. We do offer prenatal and postnatal enrollment, which has its own unique challenges and opportunities. We know that postnatal enrollment in research is really challenging. We ask parents to enroll before their child is 1 month old because of the timeliness of some of the conditions on our panel. And it's a rough time to ask people to consent to a research study. So we know that impacts our enrollment rate. The prenatal enrollment is more challenging for us, though, because you may recall that the way that we contact most of our parents is that we have the parents' contact information from the state laboratory, and we only get that contact information after the baby is born and has the newborn screening done. So you can just here get a sense of that. Our postnatal consents are the large majority of the consents that we have obtained for early check. So we ask mothers to give permission. This is a minimal risk study, so we only need one parent to give permission for their child to participate, and we ask that it be the mother. We do not have identity verification, and if folks want to talk about that during the Q&A, it's a challenge, but it's one that we can discuss more. But again, the large majority of our moms give consent via our electronic consent on a portal, and they do not engage with us unless they reach out to us. And so we have to be extremely careful that the materials that we provide them are accessible and acceptable to the state. And that's been a fun challenge. We need to make sure.
I'm not. Let me leave this then.
Oh, I think someone needs to mute. That some of the challenges we face are ensuring that we can support informed choice while not acting as a barrier to recruitment, meeting needs of a diverse population. We need to make sure that there are sufficient and feasible ways for people to communicate with a research team if they want to. And of course, there are regulatory and ethical requirements that we have to meet as a research study. So, we have all of those standard requirements that you all are very familiar with. And overall, we really want to make sure that we have an approach that's acceptable to participants and builds a sense of relationship and trustworthiness. So this is just a screenshot. I'm happy I should have included a link to the portal in this presentation, but I'm happy to send that out if folks are interested. You're welcome to go through both our consent materials and also our general education. As I mentioned before, another challenge is that our results that are normal, our negative results, are returned through the portal. And so parents receive an email informing them that their child's results are ready, and they log in to the research portal to view the actual result. They have access to a very brief lay summary as well as the actual laboratory report. And at some other time, I'd love to talk to you about the assessment data that we're collecting. We are surveying and doing interviews of participants in the Early Check project to learn both the perceptions of individuals whose baby screen negative, as well as those who are a true positive and a false positive. So that data we can talk about at a different meeting. And I'll just close by talking about our next steps. And Cindy, please chime in here. We are very pleased that we have planning grants that are underway right now for two different things. One of them is looking at chromosome 15 disorders. And then I think really both an exciting thing that we're working on, but also a thing that is quite challenging, is adding a sequencing panel. And we expect that we'll be adding a sequencing panel to Early Check by about this time next year. And I will end with some acknowledgements. Cindy, would you like to chime in?
Yeah, I'd certainly appreciate all the work, the collaborative work that goes into Early Check. Don Bailey, Holly, the group at RTI, as Holly mentioned, this is Early Check was really set up with the sort of the requirement to include CTSAs funded by NIH that are in the state. So we're fortunate to have 3 CTSAs in North Carolina. So Duke, UNC, and Wake Forest have been involved in this, as well as our partners at the state newborn lab, screening lab, as well as Scott Schoen, who heads the North Carolina State Laboratory of Public Health. And thank you to Holly for putting all these slides together.
Oh, thank you to Cindy for joining me.